Hello children. So let us talk about remaining cell organelles which are peroxisomes, ribosomes and centrosome. Along with that, we will also finish this chapter with a discussion on cell division. So, peroxisomes are membrane-bound cellular organelles and they contain mostly enzymes. But they are different from lysosomes, children, because the nature and function of these enzymes are very different. And these enzymes carry out oxidation reactions that can break down fatty acids and amino acids. They also catalyze various reactions that neutralize free radicals. Now, this is a very important aspect of the cellular homeostasis because free radicals are damaging entities, damaging chemical species which are very reactive and they can cause a lot of damage in the cell. So, to neutralize them, the essential enzymes are present in the peroxisomes. And they also are very important during the fatty acid metabolism. So we can summarize that peroxisomes are enzyme-filled membranous vesicles which perform various important functions like lipid metabolism and reactive oxygen metabolism. Let us move on to the next cell organelle, which is special because this is non-membrane bound cell organelle. Now, ribosomes, very important cell organelles. So first thing to remember, they are tiny spherical and non-membrane bound cell organelles. And where do we find them? So ribosomes are the only cell organelle which are common between a prokaryotic cell and a eukaryotic cell. So they are found in all kinds of cells, but their sizes, relative sizes are a little different. So in prokaryotic cell we will observe 70S type of ribosome, and in 80S type of ribosome will be observed in eukaryotic cell. Now what do they do? The function of ribosome is essentially protein synthesis. So wherever they are found, they will do protein synthesis. And where do we find them? So in the eukaryotic cell, the ribosomes can be found freely in the cytoplasm, or they can also be found attached on the surface of endoplasmic reticulum as we have previously discussed. But ribosomes are also found within two semi-autonomous organelles which are chloroplasts and mitochondria. As you might remember children, chloroplast and mitochondria are such cell organelles which contain their own DNA and ribosomes. So ribosomes are present in these organelles as well. So ribosomes are very essential cell organelles and the only cell organelle common between prokaryotes and eukaryotes, and they perform protein synthesis. With this, let us talk about another non-membrane bound cell organelle, centrosome. So centrosome is essentially present in animal cells only, first thing to remember. And where do we find this organelle inside the cell? It is located in the cytoplasm, usually close to the nucleus. How does it look like? So centrosomes usually contain two cylindrical structures which are known as centrioles. So one centrosome apparatus will have two tubular structures known as centrioles, and these centrioles actually lie at 90 degree to each other. So what is the function of centrioles? Centrioles perform various functions. For example, they are present at the base of cilia and flagella. Remember, cilia and flagella are the locomotory organs in unicellular organisms. So centrioles are present at the base of cilia and flagella, and they also form spindle fibers in animal cells. Now spindle fibers are those essential structures which help in the cell division by separating the chromosomes and thus sending them to two different poles so that they can become part of future daughter cells. So centrosome, once again remember, just like ribosomes, they are non-membrane bound organelles found only in animal cells, and they form spindle fibers during cell division. But here it is important to mention that even without the centrosome, found in the plant cells, they also form spindle fibers. With this, let us talk about a very important and concluding topic of this chapter, cell division. So what is cell division? As the name suggests, that division of a cell will be cell division. So the process by which new cells are made is called as cell division. So why do we want these new cells? Because every multicellular structure or every new cell has been formed by pre-existing cell. So unless a pre-existing cell divides, we are not going to have any new cells which are required in so many processes like growth, replacement of old and damaged cells, and repairment of injured portions, and also to form gametes which are required for the sexual reproduction process. So how many types of cell divisions are there? There can be two major categories of cell divisions, which are mitosis and meiosis. So let us talk about them in some detail now. So, mitosis. What happens in mitosis, children? One diploid parent cell will divide and form two daughter cells. So, the product of one mitotic division will be two daughter cells, and daughter cells will be identical to each other as well as identical to their parent cell. Not only genetically identical, they also carry the equal number of chromosomes as the parent cell. So here you can see the parent cell had 4 chromosomes, so the daughter cells each has got 4 chromosomes. Now this kind of division is most common because this is the division which takes place during the growth, repair and regeneration activities. So what is meiosis and how is it different from mitosis? Where is this required? So in meiosis, one parent cell divides and forms 4 daughter cells. Yes, one meiotic division will result in 4 daughter cells. And also, these 4 daughter cells are all different from each other genetically. They are not only different from their siblings, they are also different from their parent cell when we talk about the exact genetic makeup. Another very important aspect is that they all carry half the number of chromosomes when we compare it with the parent cell. So here parent cell was diploid, it was having 4 chromosomes, and the daughter cells are haploid and they each contain 2 chromosomes. So don't be confused that these 2 chromosomes are half. So what about the cellular activities which are to be governed by remaining half? Even one set of chromosome, which is known as the haploid set, one set of chromosome is essentially containing all the information. But for the safe side, every normal cell contains 2 sets of each chromosomes. So here the tall red has got another copy, tall blue. A small red has got another copy, small blue. Another example that I can give you to make it more clear is that we contain 46 chromosomes, but actually they are 2 sets of 23 each. So that another backup copy is always present in our body cells. If there is any mutation in one set, then the body functions can be carried out by the normal gene which is present on the other set. So the daughter cells formed by meiosis are haploid. They contain half the number of chromosomes or single set of the chromosome. That's why the name haploid. Now, where is the significance of this kind of division? Meiosis will form gametes. So, meiotic division is observed when there is requirement for formation of gametes. And you know, children, gametes have to ultimately fuse during fertilization and form zygote. So if the gamete is not containing single set, once they will fuse together, the total number of chromosome will be double. Suppose a cell of 4 chromosome is fusing with another cell of 4 chromosome, the resulting cell will have 8 chromosomes, which is not possible. So we need to maintain the chromosome number in a particular species. That's why this amazing phenomena of meiotic division has been evolved in nature, so that gametes will have half set of each uh, total chromosome, and once they fuse they will complete the set just like the parent cell. So let us go and finally compare these two kinds of divisions. But pay attention to the color of the daughter cells. That will help you in remembering that mitosis forms identical daughter cells and two daughter cells, while meiosis forms four daughter cells, and they all are different from each other. So the major differences between mitosis and meiosis. One mitotic division produces two daughter cells. Here we will get four daughter cells. All the daughter cells in mitosis are genetically alike, so they are actually clones of each other. But all the daughter cells in meiosis are genetically different from each other and from their parent cell. Chromosome number remains same after a mitotic division. So if the cell was diploid, the daughter cells will also remain diploid. But the daughter cells after meiosis contain half the number of parental chromosomal count. And the last point, where do we observe this? So mitosis is the most common division, and it takes place for growth, regeneration and repair activities. Meiosis takes place during the formation of gametes, which are the most essential part of sexual reproduction. So with this, children, we have finished this chapter. Please go through each and every video at your own pace and try to learn these concepts, revise these concepts, and augment your notes. Thank you.